Ever wondered how our cells regulate their growth? It's a complex process with one protein playing a starring role, the retinoblastoma protein, or RB. Dive deep into the microscopic world within us, and you'll find an intricate dance of life happening at every moment. This dance is orchestrated by an array of proteins, and one of the most essential among them is the retinoblastoma protein. RB, as it's often abbreviated, is a nuclear phosphoprotein. Now, that's a mouthful, isn't it? Let's break it down. It's nuclear because it resides within the nucleus of our cells. It's a phosphoprotein because it contains phosphate groups, which are crucial for its function. So, what does RB do? Its primary role is to prevent excessive cell growth during the cell cycle progression. Think of it as a traffic cop standing at the busy intersection of cell growth. It ensures that the cells don't grow too quickly or too slowly, maintaining a perfect balance. RB is not just a passive observer though. It is a hands-on regulator, actively inhibiting various nuclear proteins involved in the cell cycle. This means that RB is expressed in both cycling and resting cells. Whether the cells are dividing or taking a break, RB is always there, keeping a watchful eye on the process. One of the most crucial stages that RB regulates is the transition from the G1 phase to the S phase of the cell cycle. The G1 phase is when the cell grows and prepares to duplicate its DNA, while the S phase is when the actual DNA replication happens. RB ensures that this transition is smooth and controlled, preventing any unwarranted DNA replication. So it's clear that RB is a key player in the cell cycle, regulating the transition from the G1 phase to the S phase and inhibiting DNA replication. A deeper understanding of the intricacies of cell growth and the pivotal role of RB opens up a world of possibilities for medical science, from cancer research to genetic engineering. Stay tuned as we delve further into the mechanism of RB in our next scene. But how does RB work? Let's delve into the mechanism. The primary target of RB is a group of proteins known as the E2F family of transcription factors. These are essentially the conductors of the cell cycle orchestra, directing the timing and levels of expression of different genes involved in the process. They regulate everything from the initiation of DNA replication to the onset of mitosis, the final stage of cell division. To understand how RB interacts with E2F, we need to look at the different phases of the cell cycle. In the first phase, known as G0/G1, our cells are either at rest or just beginning to prepare for division. During this phase, RB is in a hypophosphorylated state, meaning it has fewer phosphate groups attached to it. In this state, RB binds to E2F, effectively putting the brakes on cell cycle progression. This binding inactivates E2F, preventing it from turning on the genes needed for the cell to move into the next phase. Now, let's move on to the S phase, where DNA replication occurs. If RB becomes chronically activated during this phase, it continues to bind and inhibit E2F. This leads to a downregulation of the necessary DNA replication factors that E2F would normally switch on. Essentially, RB is acting as a gatekeeper, preventing the cell from replicating its DNA and dividing before it's ready. So, you see, RB plays a critical role in regulating the cell cycle. It's like a vigilant guard, constantly monitoring the cycle's progression. And when things start to move too quickly, it steps in, interacts with the E2F transcription factors, and slows things down. It ensures that our cells only divide when they're good and ready, preventing any unwanted or excessive cell growth. This shows that RB is a master regulator, controlling the cell cycle by interacting with E2F transcription factors. To sum up, RB is a vital cog in the cell cycle regulation machine. This nuclear phosphoprotein plays the role of a stern gatekeeper, ensuring there's no overzealous cell growth during the cell cycle progression. It's like the diligent traffic officer directing the seamless transition of a cell from the G1 to the S phase and putting a pause on DNA replication when necessary. But how does RB do all this, you ask? Well, it targets the E2F factors, the puppet masters controlling the timing and levels of gene expression involved in the cell cycle process. In the restful G0/G1 phase, RB binds the E2F, effectively putting the brakes on cell cycle progression. And in the bustling S phase, a hyperactive ARB leads to a controlled slowing down of necessary DNA replication factors. So next time you wonder about the intricacies of cell growth, remember the retinoblastoma protein, the unsung hero of cell cycle regulation.